data from ZUMA-3 were presented at ASH 2017, and my guest right now back then was talking to the press and talking about how important it is that personalized cell therapy really has the potential to be an advance for adults with acute lymphoblastic leukemia and absolutely no other treatment options. So now it's ASCO, and it's time to go back again to see what's new with Dr. Bijal Shah, who is an MD and from Moffitt Cancer Center in Tampa, Florida. And let's start with a summary of what was known going into this meeting about ZUMA-3 and CAR T-cell therapy.
We saw a very high rate of MRD eradication on the ZUMA-3 trial. It was 88% when we looked at our evaluable population, and we were really excited to understand how to build on that data set. One of the things that we wanted to fundamentally understand is whether the therapies that were received beforehand would influence the endpoint, the MRD negative endpoint. We focused on Blin. Blin is another, and to be more specific, Blinatumomab is a bispecific CD3, CD19 T-cell engager that has a lot of similarities to CAR T. Can induce CRS, can induce neurotoxicity, and works by trying to drive T-cells to eradicate leukemia. Given that background, a concern emerged. Can we anticipate any meaningful benefit with CAR T-cell therapy in patients who either had previously received Blinatumomab or were just flat out refractory to Blinatumomab as they came on to the CAR T trial? We wanted to understand specifically, in trying to answer this question, we wanted to understand, is this going to be a T-cell quality issue? Is this going to be a T-cell function issue? And how do those potentially relate to the safety and efficacy with this product? And so we accordingly began by looking at the T-cell product characteristics themselves. Initially, there was some worry. We were able to effectively transduce cells regardless of prior Blina. That wasn't an issue. But we were seeing a trend towards a more differentiated phenotype. Right? So when we talk about central memory, we were seeing, again, a trend towards fewer central memory and more towards some of these effector T cells. And so there was a concern there, okay, is this going to come with A, more toxicity, and B, less overall efficacy? So as we started digging into the safety data as a start, we were relieved. We didn't see a significant increase in the rates of CRS relative to those who had never seen Blinatumomab. We did see an increase in neurotoxicity, oddly enough, in the Blinatumomab naive group. And so we wanted, again, these are small numbers. I don't want to make it sound like these are huge numbers, but we wanted to try to understand that. And as we started going back to the study population, we realized that this was confounded by the fact that there was a higher percentage of patients in the Blin naive group who received a higher dose of cell product. Because remember, this being a phase 1 trial, we're exploring different dose levels. And that probably explains that difference. Coming back to the safety data, importantly, all of the CRS and neurotoxicity was manageable. It was reversible in all patients. We didn't have any deaths from CRS or neurotoxicity in the Blin naive or Blin exposed groups. The only 2 deaths that we have reported were both related to progressive disease, with one in both groups having passed away from progressive lymphoblastic leukemia. Moving from there to the efficacy, we were again relieved. The undetectable MRD rate in the Blin exposed group was 88%, and in the Blin naive group was 100%. So we started, it was a collective sigh of relief that, okay, the Blinatumomab exposure, even in patients for whom Blinatumomab was the last line of therapy on which they progressed, the Blinatumomab exposure did not appear to imperil our ability to treat with CAR T-cell therapy. And so we went from there to actually see if we could support that, what we were seeing clinically in terms of these MRD rates, with CAR T characteristics, looking at peak and also looking at sort of the overall CAR T level over a 28-day time span. And perhaps, as you might expect, There was no difference between the Blin naive and Blin pre-exposed groups. And so for us, as I mentioned, this is very reassuring. It means that, at least in the context of Blinatumomab, prior CD19-directed therapy is not driving a difference in safety or efficacy. And I think this really does move the bar forward. I think it moves the bar forward in a couple of ways. I think, one, we have to be less apprehensive about our sequencing of therapy. And I think this may be some, I'm hopeful that we'll see similar findings with other CAR T products, you know, as more experiences emerge. But it is very reassuring to see. Are we at a point now where we can declare, okay, every patient should get Blinatumomab then CAR T? No, we're not there. You know, I don't think anyone would make those kinds of suppositions based on a, you know, a subgroup analysis from a larger phase 1 study. But it is encouraging.
So where to now with ZUMA-3?
We're actively enrolling into the phase 1. Based on the pace of accrual, it wouldn't surprise me if we were finished with the phase 1 in the next few weeks. I think once we have a final endpoint in terms of both safety and efficacy from the phase 1 study, we'll be able to pick our best dose moving forward. And I'm very excited because once we do that, we'll be able to open not just in the US but also in Europe. So we've been active in the United States now with the ZUMA-3 program, but it'll be very cool now to bring our European colleagues into the space. And again, just looking at the pace of accrual with the US sites, I don't think it's going to be a very long phase 2. I think we're going to see the trial really move very, very fast. This is a huge unmet need. You know, I didn't speak to it earlier when I was talking about our sort of the patient population that we enrolled, but these are patients who are seeing 3 and 4 and even more lines of therapy before coming on. It's very clear that we're not succeeding with conventional chemotherapy in this space, and we need to find novel alternatives with the goal of engendering meaningful remissions for these folks.
I mean, it has to be fun for an investigator to have something that seems to be working so well. And
I want to give it to everyone. That's the challenge, right? You know, we're still you know, in this era of doing this on a phase 1 study, and you know, certainly for Novartis, we do have it open on our site, and we are putting patients on the Novartis trial now that we have that as an option. But of course, I mean, it's really cool. I mean, that's usually the quote that ends up getting pulled when I do these interviews. Is it's really cool because it is.